Preterm birth, the leading cause of neonatal morbidity and mortality, is preceded by spontaneous preterm labor, a syndrome of multiple etiologies. Only pathological inflammation has been causally linked to preterm labor and birth. Inflammation is commonly initiated by innate immunity, yet inflammation can also be mediated by adaptive immunity, of which T cells are the primary component. However, their role in preterm labor is poorly understood. We hypothesized that the uncontrolled invasion of maternal effector T cells may lead to preterm labor and birth. We focused on effector T cells, since these cells are primed towards a specific antigen and can release cytotoxic molecules. First, we showed that effector T cells are present at the maternal-fetal interface and are increased in women with preterm labor. These effector T cells expressed the cytotoxic molecules granzyme B and perforin, the expression of which was highest in women with preterm labor. Can effector-activated T cells induce preterm labor and birth? To answer this, we induced T cell activation and found that the injection of anti-CD3 induces preterm labor and birth. T cell activation at the maternal-fetal interface was confirmed by the upregulation of markers such as CD69 and the release of IL-2 by decidual T cells. T cell activation induced pro-inflammatory responses in the cervix, myometrium, and at the maternal-fetal interface, and induced pro-inflammatory phenotypes in B cells and macrophages, which displayed a pro-inflammatory M1 phenotype. Yet, such changes occurred in the absence of a neutrophilic influx. Does T cell activation cause adverse neonatal outcomes? In vivo T cell activation caused a high rate of neonatal mortality. These neonates were smaller and leaner compared to controls, which was associated with a fetal inflammatory response. Finally, we asked whether T cell activation-induced preterm birth can be prevented. Treatment with progesterone reduced the rate of T cell activation-induced preterm birth and the rate of neonatal mortality, mediated in part by the downregulation of pro-inflammatory mediators. To summarize, effector and activated T cells initiate immune responses at the maternal-fetal interface, leading to preterm labor and birth, which can be abrogated by treatment with progesterone. This study was published in the Journal of Immunology and was supported by the Division of Obstetrics and Maternal-Fetal Medicine of the Intramural Program of NICHD, as well as the Perinatal Research Initiative of Wayne State University.